Hello, everybody. Greetings from Switzerland. We enjoy lovely fall weather, but already the first mountain peaks are white, and temperatures at night might very well fall below freezing. My name is Martin Wettstein, GIST patient in 2008 and member of the board of the Swiss GIST group. I'm very happy to open the next presentation and to introduce to you Professor David Josephy. He is the founder and long-term president of the GIST Sarcoma Life Raft group in Canada. And Jim Hughes, he is board member and clinical trials coordinator of the Life Raft group in the United States. Both these gentlemen have been with the greater GIST community for years, helping to navigate the science and clinical trials landscape. They are dear friends to the global community and will be sharing with us a walk through time of clinical trials and what we learned about GIST along the way. Please, David and Jim, the camera is yours.
Thank you, Martin. Is my screen sharing working okay?
Yes, perfect.
Great. So thank you very much. It's really an honor and a pleasure to be here today. And after so many years and listening to George Dimitri's recollection of the early days, it has been a long and amazing journey. And it's great to have so many people with us today. Jim and I are going to talk a little bit about clinical trials. I'm going to begin by talking in a sort of general sense about the role of clinical trials in medicine and in cancer research. I will try to keep that brief, and then Jim is going to get into the nitty-gritty details about clinical trials for GIST. Although people have been referring to me as doctor, I'm not a real doctor. I'm an academic biochemist. I'm not a physician, so I can't give anything even close to medical advice. So let me begin then by talking about clinical trials, and let's move on to the next slide if I know how to do that. Hold on. Next slide. Okay, how's that? Oh, screen sharing is stopped. Pardon me. Screen share, screen. Oh boy, pardon me. How's that? Is that all right?
Yes.
Okay,
it's not in presentation mode.
Presentation mode. So let me go to presentation mode. Oh. What's that? Okay, how's that?
Great.
Now it's fine.
So I really only know of one book that's actually on the specific topic of cancer clinical trials. It's written about 10 years ago now, but it's still a very useful guide. And I've based some of my presentation on that book. It's called Cancer Clinical Trials. And yeah, it's recommended if you are considering or interested in the topic. I think it's a pretty good introduction, mainly oriented towards patients. So Cancer Clinical Trials by Beer and Axemaker. I think it's out of print, but I think you can still get a copy on the internet. Okay, so working from that book, then they define or explain that clinical trials are experiments. They really are experiments, but they're medical experiments, clinical experiments, highly organized and complex experiments that test new therapies on volunteers. When we say new therapies, I think we're typically thinking about new drugs or drug combinations, but in fact, clinical trials go well beyond just testing new drugs. They can be testing, for example, new techniques in surgery or radiology. I think importantly for this community, they may be testing new diagnostic techniques like mutational testing, which I think we will hear more about later in the New Horizons meeting. And they can even be testing preventive methods like tamoxifen and anti-estrogens for breast cancer to prevent breast cancer recurrence. But we're going to focus primarily on clinical trials of new drugs. Beer and eggs makers point out that nearly 1 in 3 clinical trials fail to enroll even a single patient. I think that's an important caveat to bear in mind. If you look at the databases like the clinicaltrials.gov database, you'll see a great number of clinical trials, but bear in mind, a lot of those really never even get off the ground for one reason or another. They never really even get started. And about half, this is 10-year-old data, but it may not be so different today. About half of all clinical trials really never get completed simply because they do not enroll enough participants. So the general conclusion is that greater participation in clinical trials would speed the development of new treatments. Clinical trials then proceed in a number of phases, and it's worth just reviewing what those phases are in terms of the development of a new drug. So phase 1 is the initial safety testing in humans. It's designed primarily not to look at the effectiveness of a new drug, but to ask whether it's safe to use at all. So this is the first phase of a clinical trial of a new drug. Why would anybody volunteer to be, well, if you wish, a human guinea pig on a new drug? But there are, of course, motivating factors. This would be a way of getting access to the newest drugs. And there is a potential, as we've seen realized with Gleevec, there's a potential for being an early recipient of a breakthrough treatment. Of course, there are reasons to avoid a phase 1 study. There's the risk of unknown or unanticipated toxicities or side effects. And frankly, there's a generally low chance of success. I mean, a lot of clinical trials simply do not succeed. Phase 2 then is what we could call the workhorse of cancer treatment research and testing. That is the phase of testing for efficacy. Does this new drug work? It's ethically appropriate to carry out these clinical trials when there's no effective alternative treatment available. Typically, phase 2 uses a small group of patients, maybe dozens up to hundreds. As we've already heard, Chuck Court mentioned getting randomized. And that's an example of what I'm stating here, that trials are often randomized between different arms. When we say different arms, people in each arm are participating in the trial, but they'll be treated a bit differently. For example, they might get 2 different drugs or 2 different doses of the same drug. And we've already heard Chuck Court gave his example of what we call crossover. Most studies, or many studies, will involve a crossover possibility. Crossover means that if, for example, as we just heard, if the lower dose treatment is not being effective, then the patient will be switched onto another arm of the study, and that might be, for example, as we just heard, a higher dose of the same drug. The aim being to provide maximum potential for benefit to the patient. We have this principle of equipoise, which is generally applied in clinical trials. And that is that a new trial begins with what scientists would call the null hypothesis. That is, you begin with the assumption that you begin on the basis that there's no evidence that the new drug is going to be better than the existing treatments. That is, there has to be, the phrase is, genuine uncertainty over whether the new treatment will be beneficial. If there isn't genuine uncertainty, if we know the new drug is going to, or if you already know the new drug will be better, then it isn't ethical to have a clinical trial. You have to begin using the new treatment. So as the trial progresses and is monitored as it continues, it may be that the initial findings will show some evidence of efficacy. you begin to see evidence that the new drug is working. Generally, once you reach a certain predetermined threshold, statistical threshold for evidence, then there may no longer be genuine uncertainty. By this point, you may know that the new treatment is working, for example. And then you have an ethical imperative for the people running the trial to provide the superior intervention to all of the participants in the trial. These can be very difficult decisions to make. In particular, hang on. Having trouble advancing the slides. Sorry about that. Okay, let's try this one. So, I'm sorry, I should be better at this than I am. Let's see. There we go. So it does seem, what I just said may seem obvious, that if the new drug proves to be effective, once it is showing itself to be effective, then you should stop the trial in that sense and move all the patients onto the new drug. But it really isn't that simple. For example, there's a phenomenon which has been observed and commented on, that there can be a sort of lucky winning streak effect, especially if you have a small number of patients in the trial. So you may, just by statistical chance, happen to early on in the trial, you may see evidence that the new drug is working, and you may be tempted to quit while you're ahead. That is to stop the trial and put everybody onto the new drug. But there have been instances where that's been done and it turned out not to work on the larger number of patients. So there are some difficult issues to be dealt with with regard to this decision point. So the third phase of a clinical trial then is comparison, is a phase 3 trial in which we would compare the new drug to the standard of care. And this would mean increasing the scale of the trial to a larger number of patients, maybe hundreds or thousands, and doing a direct comparison of the new treatment to the standard of care. Unfortunately, even a lot of treatments that look successful in phase 1 or phase 2 trials have subsequently failed at the phase 3 level. So I'm sure there will be people present in this audience who are already thinking about whether to enter a clinical trial. Many of you are already in clinical trials. And so what does one want to think about before deciding to enter a trial, and what sort of questions should you be discussing with your doctor with regard to the risks and benefits? And here are a few examples of the questions that would be in your mind, which I can't answer, but for any particular trial, but I hope that your physicians would be able to guide you through. So first of all, what are the possible benefits? And connected to that, then what are the possible risks of the clinical participation in the clinical trial? What will happen to me during the trial? What is the planned intervention, and how will I be monitored? And what are the parameters of participation, and how long will it last, and so on. The technical questions about the way that the trial will be performed. And there is also often the question of who will pay for the costs, and the cost of participating in a trial is not only the cost of the drug itself, but you have to think about the costs of associated examinations and monitoring, and perhaps travel and accommodation costs if you have to travel to a trial center. So all of these questions should be discussed with your doctor before you decide whether to participate. I won't go on too much longer, but the last thing I want to talk about, because it's certainly a topic that's been very much in the news and certainly being discussed a lot in the Life Raft group and other patient support groups. And that's the difference or contrast between a randomized clinical trial, which I've been talking about up to this point, and on the other hand, what we call real world evidence, or RWE. So real world evidence means going beyond a clinical trial point of view and looking at what is actually happening once an approved drug or therapy gets out into the real world. So real world evidence is based not on a clinical trial, but rather on sifting through the data that has been collected from the real world use of the drug or the therapy. And that could be, for example, insurance claims or billing data to a national health service. It could come from product and disease registries, which we may talk about later in the meeting. And very importantly, it could include patient-generated data. And the Life Raft group, for example, has been very much involved in acquiring and analyzing real-world evidence from the patients who have given their data into the Life Raft group registry. An important example of this that you've heard about recently is the COVID-19 vaccine situation. All of the new vaccines have been put through clinical trials. And in the news media, you keep on hearing numbers quoted about 95% of efficacy or 50% efficacy or 75%, different numbers for different vaccines. Very important to remember, though, that as these clinical trials are progressing, each clinical trial typically has been done in a different country on a different group of patients. And while those trials are being carried out, the virus itself is mutating. We know about the Delta variant, for example. So the clinical context is changing. So it's really not one should not be surprised that the efficacy numbers that come out later from the real world evidence could be quite different from the numbers that were obtained in the randomized clinical trial, because the context is very different. And I just have one last slide to illustrate some of those differences for you before I turn it over to Jim. So if we compare randomized clinical trials on the left of the table to the real world evidence on the right. So the number of patients involved is certainly going to be larger for real world evidence once we get the therapy is being used in the general medical community. There's a difference in eligibility criteria. We didn't talk about this yet in the context of clinical trials, but it may come up later in Jim's discussion, that to get into a clinical trial, the patient has to meet what can be a very long set of eligibility criteria with regard to the general health status, the nature of the disease, and so on. On the other hand, once we get the drug or therapy out into the broader community, then those eligibility criteria are very much relaxed. For example, there was an issue with the vaccine trials that some of the early vaccine trials were limited to people under the age of 65. But once the vaccine was approved for use, then it was being used preferentially or predominantly, or a priority was given to people over 65 who might not even have been in the met the eligibility criteria for the clinical trial. So that's another dramatic difference between the way that between the results of an RCT and an RWE. Randomized clinical trials are normally being done at academic centers with very specialized doctors and treatment. Whereas real world evidence, you're getting out into the general community clinics. Compliance with the treatment may be monitored in the clinical trial, maybe not monitored at all in the real world. And the follow-up is likely to be greater in the randomized clinical trial than the real world evidence. So compared with the clinical trials, RWEs better reflect the actual clinical environment in which the medical intervention is being used, with regard to patient demographics, comorbidities, adherence, and concurrent treatments. So in that sense, the real-world evidence would seem to be superior to the clinical trial because it's really based on the real use of the drug in the real world. However, it's important to bear in mind, and the Life Raft group certainly does bear this in mind whenever we present our data from our patient registry, for example, real-world evidence is subject to biases. biases in particular because they're not randomized. So that it may be that, for example, patients who respond well may be more likely to submit their data to a registry compared to patients who don't. Or there could be geographic or educational background biases, for example. And these really can't be controlled for properly in real world evidence. So it's not that one is superior to the other. There's a role for both randomized trials and real world evidence. They both have a role to play. So at that point, I'm going to stop and turn things over to my colleague, Jim Hughes, who's going to talk more specifically about particular clinical trials for GIST. And I guess we'll leave questions until the end. Is that right?
That is correct. Do you mind unsharing your screen so Jim
I
can
will
post
now
it up?
stop sharing. There we go.
Thank you.
Okay. First, I have to go into slideshow mode. Can everybody hear me?
Yes.
Yes.
Okay, hang on a sec. Okay, and then I need to share this. Okay, hang on.
Very good.
I'm not sharing anything right now, am I? I just
No.
lost my screen here. Hang on a sec. There we go.
I can see you.
Yeah, I know. Okay, I'm going to share screen 2 here. There we go. Can everybody see something about clinical trials with my name on it?
Right.
Okay, I have to pull up another screen here. Hang on a sec. There we go. That's the screen I want in front of me. And that's the screen you're going to see. Okay. So you should see the New Horizons GIST screen and just that. And I should be looking at the previews and everything. Does everybody hear me okay? I hope. Oh,
Yep.
Sarah, would you please watch the chat and let me know if I get a question? I'm not a good multitasker at this
Oh,
age.
no
for some
problem.
reason.
We're going to save, there were some questions that also came in for David, so we'll say
Okay.
them. Please
right.
save some time at the end for questions.
If it's something about how I'm doing this and you can't hear me or something's not clear, please interrupt me and I'll try and fix it. Okay, again, my name is Jim Hughes and I am here in Illinois, near Northwestern University in Evanston, just north of Chicago. Margie and I, the parents of Nancy Hughes Welsh. Nancy was our daughter, and this will be in memory of Nancy. Nancy was born in 1967 and passed away due to GIST in 2005. My disclaimer says I'm not a physician. I am nothing close to that. David has some background in biology and chemistry, and I have none of that. I worked for the phone company for 30 years in planning and engineering. So please take everything I'm saying to the doctor or get a question asked about anything I'm saying. I just want to talk a minute about my daughter, Nancy, and I need to advance my slides here. Hang on a second. Oh, I'm not doing it, so I got to figure out how to do that. There we go. I took this somewhere towards the end of high school, about 1985 or so, in our front yard. And Nancy, normal childhood, nothing to say anything like what happened to us eventually happened, was about to come. It was in 1991. When Nancy was 23 years old and had been working for a little while, that she woke up in the morning and was throwing up blood. And we went to the hospital, and long story short, she was diagnosed with GIST in her stomach. It was 4 tumors. She was 23 years old, half the stomach was removed, and she immediately began a series of chemotherapies. This was taken out in front of our house, and we got a new car, Nancy did, to go to chemotherapy. Basically, she was going to be making a lot of doctor's appointments. This is a cache we found much later. This is like real world evidence. This was all the bands from all the hospitalizations. Nancy, for some reason, was just taking them off and putting them in a bag in her closet. And so we found this after she passed, and we immediately recognized how much and how many times we had been with her at the hospital. We thought it was something like 36 hospitalizations. There are about 52 bands here. So it just gives you some idea of how intense this was for some period of time in 1991 till 2001. And I have to say, listen to Chuck, her story is very similar. Leiomyosarcoma was the first diagnosis, and we had lots of chemotherapy. I think there were maybe 6 to 8 chemotherapies. That did not work. It recurred about every 3 years. So in 91, surgery, 94, surgery. Again, I think in a little sooner now, about 97. Nancy married in the middle of all this, and basically we were just managing this by doing surgery. And it wasn't until 2001, when she was starting to have another recurrence, that another doctor saw her and had recently heard about a patient who had what was now called GIST and had been treated with imatinib. So he sent her tumor out to be stained, and it came back c-KIT positive in 2001. And we learned within a matter of a week, Nancy had no longer had leiomyosarcoma, had GIST, and that there was a miracle drug available for her treatment. It was at that same time that we got on the leiomyosarcoma LMS listserv and began reading the same things that Chuck talked about, the stories about patients who were trying this drug and experimenting in trials with this and reporting side effects. And we just learned just in a matter of days, 1 or 2 days, everything that probably anybody really knew about just in that very short time frame. And we're somewhat prepared when the doctor told us, we're going to put you into the phase 3 trial for imatinib. Unfortunately, Nancy could not withstand Gleevec. And after bleeding out 3 times, the third time, the most severe time, resulted in 6 days in the intensive care unit here at the local hospital. There was no longer going to be a clinical trial for Nancy. So we left the phase 3 clinical trial and began a period of experimenting to see if we could find a dosage of Gleevec that would work. We never did find one that would work. And again, she bled out at 600 milligrams. And so we stopped Gleevec, and in 2002, a long period of looking for clinical trials, because now we were familiar with a clinical trial. We had been in a phase 3 clinical trial, the imatinib trial. And we had met, we hadn't really met anybody at Dana-Farber, but we had put feelers out to find out about what's next. And at that time, there wasn't anything, and sunitinib was in phase 1 and beginning phase 2. Nancy did not qualify for phase 1, and we got in very late on the tail end of phase 2. And we moved to Boston, where we lived for 3 months with Nancy. Her case was such that she could not really do this from home and manage it remotely by flying. So we lived there, and one of us, my wife or I, were there with her the whole time she was there. It was about 3 months in 2003. This next photo is from ASCO 2003. After having gone through the trauma of that phase 3 trial and having wound up in the intensive care unit, we were somewhat gun shy about trials. And it was at this ASCO that Brian Drucker spoke about imatinib and CML. Nancy's doctor came by our booth here. We're at the Life Raft group booth, and this was really my introduction to the Life Raft group. And her doctor grabbed me and grabbed Nancy. We have her right in the middle of between the two of us. And he said, we don't have passes, but we're going to walk through the door and we're going to hear Brian Drucker speak. So we just barged through, and we went into the keynote speech, and we listened to Brian Drucker. It was really exciting to hear and watch the reaction to what was now a famous drug and was on the cover of Time magazine. And this somewhat reinvigorated us, and we began looking seriously into this phase 2 clinical trial for sunitinib, and Nancy managed to get into that trial, which we'll talk about a little bit here in the next couple of slides. This is the last picture I have of Nancy. It was taken April 9th, and it was 3 days before Nancy had a bleed, and her death was the result of a tumor bleed, which she'd had many of, and this one was much more serious. We went to the emergency room, and there they intubated Nancy, and she went immediately to the intensive care unit. And it was 16 days in the intensive care unit, and after major organ failure, we lost her. So we were doing okay. Nancy was managing a large tumor burden, and she had had a significant response to imatinib. We found out, I'm sorry, to sunitinib. It was a 30% reduction in some of the sentinel tumors they were monitoring. We found out much later in 2014, after having Nancy's tumor retested, that she was SDHB deficient. And while we did not find any SDH mutations on mutation testing at that time, we suspect that Nancy was SDH epimutant. And because we didn't know this at the time, we were rather fortunate to get in the sunitinib trial because it did shrink some of her tumors and it bought us time. But Nancy had this problem with bleeding, and it had been a recurrent theme throughout our history with first leiomyosarcoma and then with GIST. So we talk about what questions to ask about clinical trials because there are a lot of issues to ask about if you're considering a clinical trial. But the most important question is to just ask the first time. Can you help me develop treatment plans, including clinical trial options if appropriate? I think we would improve recruitment and availability and the time we complete trials in if patients were more aware of this and approach their physicians up front to say, you know, when we're discussing my disease, are you capable of getting me a clinical trial option if I should need one in the future? Might not be appropriate now, but just bring it up, to bring it up and open the discussion and find out what's the position of this institution and with this practice. Do you use them? Do you recommend them? Do you work with doctors who are principal investigators? Do you have patients who are working or in clinical trials with whom you help them with their clinical trial process? So I think that's a very important point to make. Just ask. Okay, I'm going to talk a lot about trials, and I have a lot of data. I manage the Life Raft Group's database, where we scan for new trials and we look at the status of trials and we update sites. And we have well over 200 trials in our database, mainly gathered from the National Institute's clinicaltrials.gov. In getting ready for this presentation, which I'd done before for a group here in the US, I had focused mainly on the US trials and what was available here. And I knew I needed to include more of the trials outside the US that would be in these other databases. So I began to look again at some of the outside databases. And the European Union has a database. The UK, there's a company that used to be called Biomed Central, now called BMC, has the ISRCTN database, which is largely, I think, UK plus other trials around the world. And it, from what I understand, a lot of the trials there that are entered there are duplicated in other registries. And there's also the World Health Organization database, which is a good database to find out about trials in many countries that aren't typically seen in clinicaltrials.gov. So I combed through these and started looking to see if I had missed anything. Were there trials outside the US that just didn't get entered in our database? And I found that a lot of trials are double listed. And because of the conventions of how to report trials, some are still listed as authorized, but they have been completed a long time ago and they're no longer recruiting. And so this became a process of just trying to weed this all out and figure out where we still had trials running and what was available for GIST patients. So one issue I find that we need to address going forward is how to rationalize all these different databases and get the information in one place. We're trying to do it. We have our own database specifically for GIST trials. And as I mentioned, it's from the NIH registry. And it includes trials that do not specify GIST, but have relevance in GIST. So you'll occasionally find a trial that says it's for soft tissue sarcoma or solid tumors. And really it's mainly GIST. And you don't know it until you begin to look at the drug and more about how it operates. And you see they definitely mention GIST in the inclusion criteria, but they don't include it above as a subset of diseases. So you have to dig and you find that you have trials out there that do fit that pattern. We do not include all observational studies. we're mainly focused on therapeutic. If it's an observational study that might have impact on changing the management of GIST, we'll put that in there just to document it, and also to put the references in there, because we include links to trial reports and drug-specific web pages about the operation of these drugs. There's a lot of information with a phase 1 trial that is in some of the new drug development journals, and we'll try and pick up some of those to get some idea of what other targets the drug hits, maybe a little bit about what side effects were observed in some of the animal models, things like that. So you can find that at https://gisttrials.org. And I think we did this, we walked through this earlier, and I'm not going to go over that. This is just to keep in mind that we do have trials with placebos, because I'm going to talk a little bit about that in phase 3. Okay, this is a very busy slide. We're not going to talk about all these. I found that we had, when I looked backwards in time, because we were going to do history here, that I could find, if I screened on phase 3 trials and looked at drugs that had FDA approval, and then ones I remembered that had been in trial, I came up, by going through that process, with 25 phase 3 trials that related to drugs that were on their way for approval, or were in the process of being evaluated for a second indication, or being modified in some way, maybe to go for a second line instead of third line, now that they had third line, or trying for equivalence, so functional equivalence. And so there are a couple of things on here that come out. First of all, we've had a lot of trials in GIST. Everything from imatinib to now the most recent one in China, famitinib, which is a phase 3 trial. There is one phase 1 trial in here. All the rest are phase 3. And that's the avapritinib trial marked in yellow there, D842V. Avapritinib was approved for D842V GIST on the basis of a phase 1 trial. It's also worth noting that imatinib was approved for GIST based on the results, the objective response, in a phase 2 trial. I don't have that trial here. It was 147 patients. Probably should have put it here, but I really wanted to get the last phases and what our participation was. And when you look at the bottom line here, if you can see the number at the very bottom, 7,907 patients. Sounds very exact. It's really probably around 8,000 patients. This represents about 40% of all the patient accruals numbers we have in our GIST database. But it's significant in that all these patients here contributed to what we today have as a complete hand in managing GIST. I wouldn't say complete, but I would say a far more complete than many other diseases. It's certainly made a difference here in our lives. And I know it's made a difference for those who had leiomyosarcoma and were, like Nancy, diagnosed and have the opportunity to fight this disease with multiple options now. There are fourth line drugs available for this. So I wanted to be sure we recognized, and we don't today really have a way of recognizing patients. And I wanted to point out that there are a lot of patients still today who've been through trials who we really should recognize in some way, because they have served everyone here just by participating in it. Maybe the trial, maybe they've passed on, maybe the trial wasn't successful, but everybody here who participated helped solve a question, answer a question about GIST. The other thing you'll notice in here, and this came up earlier, David mentioned it, is that we have a number of trials that are terminated in the status. This happens for a number of reasons, primarily low accrual. And that happens, as it turns out, when we look at it, because in some cases, these trials are not positioned well with the design and where they're being trialed. So the case of the one trial I have there, the one that's marked SARC 2019, That trial was positioned at sarcoma centers across the US, but it was primarily for patients who were initially starting out on imatinib. It was to help determine what's the best dose using therapeutic drug monitoring. And it only accrued 5 patients over a year. And simply because at the major sarcoma centers, patients usually don't start on imatinib there. They come there after they've had progression. So that was not well positioned, and it was terminated by SARC because of low accrual. Nilotinib 800 and imatinib 800 was terminated by the sponsor. At this point, Novartis had a number of trials in nilotinib that were not successful, and they decided not to pursue that any further. So there are other reasons for doing this. There is one trial, IPI 504, that was terminated because of adverse events. And this doesn't often happen, at least in GIST, but here it's a good thing because we have a process where we're constantly monitoring safety against efficacy. And in this case, there were some deaths due to liver toxicity. And this was pulled. So it was a very important point to make that trials are risky. They do cause adverse events, and in some cases, you will find that they'll be terminated because of that. So a question that comes up with clinical trials when you're evaluating them is, will I be able to get this drug if I'm benefiting? Should the trial be terminated or after it's completed? And this is an issue sometimes with clinical trials, that the agreements that we have that you have to sign, the consent forms, need to be clear about how you're going to be able to access the drug should you be doing well on it after the trial ends. So that's a really good question to ask when you see this data. The other thing you'll note is that we started out with a lot of other industries, other than industry sponsors. And other usually means a major medical center or a consortium. The NIH NCI is noted here. It's not in the other category, it's separate. And we've only had 2 trials that were supported by the NIH. The adjuvant imatinib trial, that's noted there, the 3rd from the bottom, and also the imatinib plus bevacizumab. And that was terminated even though it was widely promulgated across the US, I think hundreds of sites, because it just wasn't in the patient's best interest. At that time, they could get imatinib. And so the question was, would you like to participate in a clinical trial where we're going to take a drug you really like and really has a great chance of performing well for you, and we're going to add another drug that might add some side effects to that? And most people would say to that, no, I don't think so. I think I'll just go with the vanilla without the chocolate sauce on top. And that's what happened. It just wasn't an attractive offering for patients, and so it was eventually terminated. And it was reported on. So you can see on the far right, another point that we want to make with this slide is that a number of these trials now have results posted in clinicaltrials.gov, and this is independent of the papers that are printed in the New England Journal of Medicine, the Lancet, Journal of Clinical Oncology. This is where industry has been asked to come back and make sure that we know more about clinical trials results and to be transparent. So you'll see here, even those that weren't successful have reports, and there is data in there that's helpful in some cases to understand how the drug performed in terms of this particular subset of patients, what were the adverse events, and were there any long-term responses, how many were there, things like that. Did they meet the objectives, and what were those? So this is really a good thing for patients to have because it provides sort of a standard way of comparing trials. And so we encourage the drug companies and anyone running a clinical trial to post these results. And part of our database includes the ability to link to these kinds of reports when they're published. Okay, this is the scenario, 25 trials. So I wanted to get a picture of what did that look like if we graphed it out in terms of how many patients were accrued over time. And so this is those 25 trials, and I've put here on this same graphic the points where there were approvals. So the start dates are where I have these accruals calculated and accumulated. So that's really sort of an artificial way to do this. But I wanted to get a feeling for over time. Was this a constantly growing? Was it declining? And what I found out is with this trend line is we've had a very fairly steady continuing accrual at this level in phase 3 GISTs for advanced GIST and for even second line and even trials at first line again. Some successful, some not. So this is 20 years of GIST accruals at the phase 3 level. And you can see that we had a very flat spot from about 2009 until about 2017. And that doesn't seem uncommon. There was another one from 2004 to about 2008. And approvals rated about every 3 to 4 years here. So we've had imatinib, sunitinib in 2006, 4 years later. And then 7 years later, regorafenib, and then 2 drugs approved in 2020, ripretinib in May, and then avapritinib for D842V GIST in January of 2020. I also noted on here the first 1 year adjuvant imatinib in 2008, and the 3-year adjuvant imatinib in February of 2012. And all the patients who were accrued for all the trials that got these drugs approved, and of course the ones that weren't approved, are here. So same number as it was in the previous slide in terms of the total number. So where were those trials held? I thought it'd be helpful just to get an idea of where clinical trials today are being run. And you can do this if you take and put all the data into clinicaltrials.gov. You need the NCT numbers. And if you load a search of all those 25 NCT numbers, you can get a picture of where all the sites are. So this is the global view of where all the trials were run. And now, so there were 16 trials in the United States. That means there are many sites, but altogether they comprise the 16 trials. And the same thing in Europe and the EU, 10 in Southeast, well, not Southeast Asia, in China, I think, and Korea. And then in Malaysia and Australia, 6. In South America, there were 3 trials that were on trial there in this set of phase 3 trials.
Jim, we have only a couple minutes left, okay?
Okay. All right, so just going a little bit further, I did the dive on where they were by state, in the European Union where they were. And you can see here they're mainly in the UK and the European Union countries. And again, one more dive into South America. These are the current trials in GIST. And I wanted to show this to just give you some idea of what is out there today. I think there are 19 here, and then the immunotherapy slide coming up, there are like 4 more. And I'll only mention a couple here. There are 2 new ones at the top in green. They're not recruiting yet. Ripretinib and binimetinib, and imatinib 10 years in France. And the ripretinib binimetinib will be an international trial. It will be in Miami here at Sylvester, Memorial Sloan Kettering, MD Anderson in Canada, and in Essen, Germany. And it's being run by Decipher. We also had a drug called DS6157A that I did this in August, and since then it has come off the list. It's no longer recruiting. And I wanted to point it out because it's a very interesting new approach. It is not following the standard KIT signaling path approach to inhibiting cell growth. And what this drug does, it attaches to a cell surface protein that's common in GIST. And it brings with it a package of chemotherapy. And so it's like sort of putting an explosive, plastic explosive on the side of a safe. It only hurts the safe. So it does, has a very interesting approach. It's different. It would work against any GIST type, including SDH and NF1. And so we're watching its success in other diseases and wondering if it will be successful here. And what's happened is this trial has been moved back now. to accrue only 34, and its primary date end date has been moved up. So I don't know what the implications of that are, but we are watching it and asking. We wrote a letter to Suzanne George, and she is asking Daiichi Sankyo if they could comment on why they're doing this, because I think it's under their control as far as press releases go. This is the picture of what drugs are approved for immune therapy. And so you have some lingo to work with here and understand what's available coming up here in the next slide. And these are the trials that are available in immunotherapy in GIST. And the one at the top there at UCLA has just been put on active but not recruiting. And I suspect it's because it's right at the end. It's probably accrued all it's going to accrue. And we'll see the final report on that. There's been some interim ones. There's been some interesting results, but I don't think there's been anything truly groundbreaking yet in this area. And there are a number of trials now in Europe, in Poland, France, and also in South Korea of immunotherapies that are available. So where are all these current trials? This is the same view, the global view, where the sites are. Again, a very coastal view in the United States and also in the Midwest, where most of these trials are run. In Europe, in South America, only one trial. Brazil, Argentina, Venezuela, I believe. So I have to stop for a minute and just say, how much time do we have, Sarah? Are we out?
We are really at the question and answer.
Okay, I didn't realize where we were here. So I'm going to just mention one thing here about clinical trials, and that is during this COVID interval, we have seen a lot of patients visiting trial using telemedicine, getting managed in clinical trials via telemedicine. And that's beginning to tighten up. And there's a database online. You can find out what states allow doctors who don't work in those states, not licensed in those states, to be able to waive the requirements and be able to see patients through telemedicine. And so this is a situation that's both tight and loose. It tightened up after before the COVID Delta variants really took hold. And then it may be loosening up again as we go through finish up with the COVID Delta variant and the wave that came through. But what's happening is states are opening and closing their states of emergency. And as they do that, these waivers are available during the states of emergency, and they're not. And this database tells you what states will allow doctors who are not licensed in that state to do telemedicine in that state. And this is the data for Illinois, so you can get some idea about the details. I'll stop here and let's go to questions. I didn't see if we had any, but there's a lot of data. Is there anything I can answer, Sarah, that you have seen from the audience?
Yeah, why don't you unshare your screen, open it up to
Sure.
the participant view, and then Martin, why don't you go ahead and start with the first question?
Okay, anybody questions? We have a chat here.
There are some in the chat.
Yes.
Martin, maybe I can start with this, a question from Carolyn DeVault.
Okay.
Does crossover, and Jim, I can bring you in on this, I think.
Mhm.
Does crossover in a trial apply when there are two drugs being studied to determine which is more effective? In other words, if a trial participant is on one of the drugs and cancer progresses, will the individual then be given the other drug under study? I think you showed an example of that, Jim, didn't you?
I believe there have been. I can't point to one
Not
right now.
commonly, but there could be.
Yeah. More often we have had placebo-based trials. In one of those earlier slides, I showed how many we had with placebos. I think there were like 7 out of 24, 23. So about a quarter of our trials, a third to a quarter, were placebo-based.
But there have been trials in which a new drug challenges an existing drug.
Yes, there have been, as you saw on
and those
that
allow
list.
crossover, I think.
I believe they do. It depends on, I think it depends on who the sponsor is and what access they have to both drugs.
So I guess the answer is yes and no. It just depends
Yes,
on the.
right. You have to check. Certainly a good question to ask about crossover is, will I be allowed to do it? And will I be monitored for progression on a regular basis in order to determine when it's time to make that crossover point?
And Martin, could I move on to the question from Yvonne about if Gleevec stops working?
Yes.
So Yvonne asks, if Gleevec 800 mg stops working, would a phase 1 or phase 3 be recommended or simply move to Sutent? It seems that the risk is high if you've only been on Gleevec to move to a trial. Thoughts? And I mean, I think generally, Jim, isn't it the case that you would not be entering a clinical trial until the approved lines of therapy have failed? I mean, I don't think you would move anyone directly from Gleevec, even at 800 mg, onto a clinical trial. Would that ever happen?
It would. It is happening actually with ripretinib and sunitinib. Ripretinib, Decipher, has a second-line trial of ripretinib, and it's seeking patients who failed imatinib and are eligible to enter that before they've had sunitinib, and they will get either sunitinib or ripretinib.
Right. Okay.
And we note that some trials today do not specify that you must fail all 4 drugs. They say if you fail imatinib and or sunitinib, that kind of language we see in the inclusion criteria. So there are doors open to second and third line. It's a question of, in some cases, can you, so for instance, sunitinib is looked at as a fairly toxic drug, and ripretinib has a different side effect profile. You might benefit from taking a chance on that if you thought that you'd like to try a chance at avoiding the side effect profile of sunitinib. You might go into a randomized trial to get ripretinib.
That's a situation where I think you'd really want your doctor's careful advice on whether to proceed onto the approved second line or to try a clinical trial with a different drug. It must be a tough.
I recently helped a patient who is going from imatinib to regorafenib, skipping sunitinib completely, and the reason given was because of the side effect profiles.
So, important question.
There's a question of Cheryl. In the interest of that collaborative approach we've talked about several times, is there anything we can do to help permanently remove those state? I just lost it. Is there anything we can do to help permanently remove those state licensing barriers to telehealth and trials on treatment?
That's a really good question. I think as we proliferate the higher bandwidth cellular phones and the broadband more generally, that there will be greater opportunities to manage patients of all types, cancer included, remotely. I will tell you that my daughter, Nancy, did go to Dana-Farber, and when they evaluated her, they told her, when they accepted her for the trial, that she looked better in person than she did on paper. So I don't know how I look on this TV screen, this Zoom meeting, but in some cases, you're probably better off actually going to be seen. Maybe not, but the idea that we can use that going forward is a real game changer. What we've learned through the COVID experience is that there are ways to manage things remotely, that it makes for a more collaborative environment with the local primary oncologist, with the principal investigator remotely. And it means that we be able to access patients and allow patients to participate in trials who could not travel. Our daughter could travel and we could travel, and we were very fortunate in that regard. Many patients can't do that. They can't stop and drop work and family and take a week off or 4 weeks like we did. So it's imperative that these alternatives be looked at, and we look at these rules. And the way things are managed here in the United States today is every state has its own authority to license doctors, and the rules don't vary that much. But there is this barrier between states. You must, if you're going to practice in Illinois, you need to be licensed in Illinois. Same thing in Florida. So if you want to participate in telemedicine, it's your local state that governs what is available in terms of how you can do things remotely. And many of these require that you have at least one established visit with your physician, remote physician, before you can begin to do telemedicine. So things are changing, and they're vacillating right now. And I think it's a good point for advocacy to take up and to try to move forward to begin to open the door to continued remote management and access to clinical trials.
Okay, thank you very much. Well, I think that's the end of the session. Thank you very much, David and Jim, for this comprehensive and profound information. You really became self-made experts in these years. It's so helpful and interesting to look behind the curtains, and New Horizons is really the perfect chance to get such information to the patient community. Thanks again, and all the best to you.
Thank you. So at this moment in our schedule, we have a 15-minute break. We are going to return again at 11:15 so we can hear from our friend Giora Scharf from the CML community and Norman Scherzer on lessons learned from the CML and GIST communities. So we will see you shortly. If you would like to chat with one another during the coffee break, as we tend to do during coffee breaks in person at New Horizons, you're welcome to do so.
Sarah, for those who are still listening during the coffee break, I did want to go back to one of the questions that was asked.
Sure.
There was a question, is it safe to try natural cancer remedies, such as mushrooms, for example, along with imatinib? Or are you risking interference with the effects of imatinib? So I just wanted to comment on that of course, a lot of patients do try natural product remedies, certainly very commonly done. I think my advice would be, it's very important to discuss that with your physician. We know that there are natural products. I mean, one of the well-known ones is St. John's wort that are well known to interact with imatinib. They do have an effect on the pharmacokinetics of imatinib. So I think it's very important, if you are taking natural, I'm not advising people against taking natural remedies, but the one thing I would say is be very sure that your doctor knows about it, because there are indeed some possible interactions.
You see, we had a question about from Yi. If I understood right, ripretinib is on a clinical trial in South Korea. Ripretinib is the fourth medicine after Stivarga, ? And ripretinib can replace Sutent also? Those are two questions. I don't know where it is in approval in South Korea, but here in the United States, ripretinib is approved for fourth line. It is also in trial for second line. And we're waiting the outcome of that trial, which could affect the order of drugs, wherein ripretinib would be the second line drug. Ripretinib had such promising results in its early trials that it looked like it might have a greater progression-free survival than sunitinib, even in the third and fourth line. So it looks like a natural to try it at second line, and that's what they're doing. And that trial has stopped recruiting, so we're expecting to hear about that in the near term, or by early next year, I think. Thanks for the question, Yi.